All nucleic acids have the same three basic components: a phosphate group, a 5-carbon sugar, which is deoxyribose in DNA and ribose in RNA, and a nucleotide base. In DNA, which has a double helix with 2 strands, the nucleotide bases bind to each other using hydrogen bonds. RNA, on the other hand, only has a single strand and therefore is a single helix. There are 2 types of nucleotide bases: purines, which have a 2-ring structure, and pyrimidines, which have a 1-ring structure. Specific purines, adenine and guanine, bind to specific pyrimidines: thymine, uracil, and cytosine. Adenine only binds to thymine in DNA and uracil, which is only found in RNA. Guanine binds to only cytosine.
The manner in which DNA replicates is known as semi-conservative because each newly synthesized strand of DNA consists of both one parental strand and one new strand.
In the first part of polypeptide synthesis, the DNA must be transcribed into mRNA sequences. In order to do this, the DNA opens up a small bubble that contains the preferred gene, and RNA polymerase can bind to one strand of the double helix DNA. The DNA is read in the 3' to 5' direction, and the mRNA is synthesized through RNA polymerase by adding bases to the 3' end. Once the preferred gene is fully transcribed, the bubble closes, and mRNA can leave the DNA. In eukaryotes, this mRNA may be further processed, while in prokaryotes, the mRNA may be ready to use in protein synthesis.
In eukaryotes, the mRNA transcript is modified before it enters the cytoplasm. This process is called RNA processing. The 5' end of the pre-mRNA molecule receives a 5' cap or a modified guanine nucleotide added after the transcription of the first 20 to 40 nucleotides. Polyadenylation occurs at the 3' end, in which 50 to 250 adenine nucleotides are added, creating a poly A tail. Both of these caps serve to protect the mRNA molecule when it enters the cytoplasm. RNA splicing is yet another part of this process and involves the cutting away of intervening sequences or introns from the final RNA. Such sequences are ultimately not translated, while the exons or the sequences that remain in the mature mRNA are translated.
Once the mRNA has been processed, it is ready for the sequencing of a polypeptide sequence. A ribosome is constructed from a small ribosomal subunit and a large ribosomal subunit and rRNA, which can then read the codons, triplets on the mRNA for the synthesis of the polypeptide. The mRNA is read by starting at the 5' end and moving towards 3' end, and tRNA matches anticodons to the codons on the mRNA to bring appropriate amino acids to bring and build the growing polypeptide. There are 3 sites on the ribosome. The first is the A site, which is where the tRNA attaches and brings in a new polypeptide. The tRNA then moves into the P site, where it takes and holds the polypeptide sequence and adds its own amino acid to it. The tRNA then moves to the E site, from which it can exit and gives the polypeptide sequence to the incoming tRNAs. This continual process of building the polypeptide sequence contributes to the overall growth of the protein, and once the mRNA reaches a stop codon, the synthesis of the protein or polypeptide is complete.
In order to activate an amino acid sequence, the enzyme amino acyl tRNA synthase must be used. An amino acid binds to the enzyme with ATP, which then loses 2 phosphate groups, making AMP. tRNA then takes the place of AMP and binds to the enzyme. Then the enzyme activates and releases amino acyl tRNA, or an active amino acid.
A codon is a triplet of nitrogenous bases found on messenger RNA that code for specific amino acid. An anticodon is a complementary set of nitrogenous bases found on tRNA. Anticodons are antiparallel to codons.
DNA replication begins with the cleaving of hydrogen bonds between nucleotides and the untwisting and separating of the 2 strands of the double helix by helicase. A replication bubble is subsequently formed, with replication forks at the ends of the bubble. Single-strand binding proteins then bind to the unpaired DNA strands to prevent them from repairing. Next, an enzyme called primase attaches to a strand and creates a primer composed of RNA, which allows for DNA polymerase to bind to and continue synthesizing the new strands. Because DNA is antiparallel, there are leading and lagging strands. The leading strand moves towards a replication fork and is uninterrupted because then DNA polymerase 3 can work in its 5' to 3' direction without difficulty. In contrast, the lagging strand moves away from the replication fork. Because DNA polymerase has such directionality, it must synthesize short fragments of DNA called Okazaki fragments. DNA polymerase 1 then replaces the RNA primers at the start of each fragment with DNA nucleotides, and ligase subsequently connects the fragments. Another enzyme called DNA polymerase 2 moves along the strand in the 3 to 5' direction and proofreads the newly synthesized strand.